So hello and welcome to episode 13 of CCLG's Research Talks for Parents and Public. I'm Ellie Ellicott and my job is to share research with our supporters. So starting with some housekeeping, we'll be holding a Q&A session at the end of the talk. So please do submit your questions whenever you think of one using the Q&A button that's at the bottom of the screen. If you'd like to use the chat function, please make sure you set the to drop down to everyone or people may not see your message. This webinar is being recorded and will be available publicly on our YouTube channel, so we will send out the link when it's ready to watch. So this month's talk is called High Might Immunotherapy Improve Outcomes for Bone Tumours and is being led by Dr. Jonathan Fisher. Jonathan runs a research lab at University College London and treats teenagers and young adults with cancer as a doctor at the University College London Hospital. His research looks at how to engineer immune cells to fight cancer. In his Little Princess Trust funded project, funded through the CCLG Research Funding Network, he is working on new immunotherapies for osteosarcoma. So, without further ado, I'm going to hand over to Jonathan.
Well, firstly, thank you very much for the invitation to speak. It's really wonderful to be here this evening. And as you said, I'm going to give you an overview of some of the work that we've been doing in the lab and some of our findings that we think are looking quite promising. But really, to start at the beginning, let's start basic. So, let's talk a little bit about the immune system. So the immune system is one of the most powerful protective forces that the human body has. And it protects us against infections. It protects us against harmful substances in the environment, things that might cause irritation, and that could be anything from pollen to asbestos. And it also continuously provides surveillance for disease-causing changes in the body, which is ultimately what leads to cancer. Now, the issue is that once you've got a cancer, that system has failed. And what happens is that the cancer then continues to suppress the immune system and leads to this kind of vicious cycle, and you end up with the development of a tumor, of a solid lump. Now, there's been a variety of different strategies used to help prevent that from causing disease or help to treat disease once that has already happened. So, and this is called cancer immunotherapy. And that was really, as it says on the tin, it is harnessing the power of the immune system to attack cancer. Now, there's really two ways of doing this. So, first way is to use what is already there in the system. So, the immune system normally responds to the presence of a tumor. And, the tumor will be infiltrated by immune cells that will sit there. And you'd think, well, if the immune cells are going in there, why isn't the tumor just killed? Well, the cancer essentially puts the brakes on the immune system, which means that the tumor cells evade detection or evade killing. And then you end up with the generation of a lump. Now, what you can do is prevent the cancer from putting the brakes on the immune system by using molecules called antibodies. And, there's a slightly more sciencey diagram here, but basically what you do is you prevent interaction between this thing called PD-1, which is a molecule that you see on the immune cells, and this thing called PD-L1, which sits on the cancer. And the PD-L1 is essentially the foot that pushes the brake pedal. If you block that, you take the brakes off, and then the cells that are already in the tumor can start to attack the cancer. And this is something that has worked remarkably well for some tumors, such as melanoma. It's completely revolutionized melanoma treatment. And it's something that is, the guys who invented this ended up with Nobel Prizes because this was really. Now, the other way of doing it, and this is more what I do, is to design something new. So there are lots of tumors, and especially in children and in young people, where the immune infiltrate into the tumor is not quite so great. So you really need to boost the immune system in a new way. So rather than trying to interfere with these processes here, you design some new pieces of immune machinery for maximum effectiveness and then deliver them to the patient. Now, some of you may have come across a type of treatment called CAR T cells. It's something that's received quite a lot of press in the leukemia and hematology field, and it's been highly successful for treating leukemia. It has been less successful in the treatment of solid lump-forming tumors. And there's potentially a few reasons for that. But really, one of the things that I wanted to do and my research group wanted to do was address the fact that that hadn't really worked. So we wanted to go back to the start to design something specifically with solid tumors in mind, and in this case, with bone-resident tumors in mind, such as osteosarcoma. So really, it ends up being a little bit like, as I say, biological Lego. So we have the ability to build new pieces of machinery to make immune cells do things that they wouldn't normally do. And this is modular. So it is literally like sticking things together to build a new Lego model. And you can work out what you want it to do based on the pieces that you put in. So I'm going to use this metaphor as we go through because I find it quite a helpful way to talk about the engineering that we've been doing. So, if you think about what you might need in order to get an immune cell or a bunch of immune cells to go to a tumor and kill it and make sure that they stay killing it, there's going to be a few different things that are required. So you're going to have to get the cells to the right place. There's no point having a tumor in your leg and a load of cells in your arm. It's not going to help. Once it gets there, it has to stay healthy. So, the tumor environment is hostile to the cells. So it has to the immune cells have to stay healthy. They have to remain with that kind of immune power to be able to kill the tumor. They have to be able to deliver whatever that kill signal is. So the cargo that allows the delivery of the kill signal needs to be powerful enough to be able to get the result that you want. And then as you're doing that, you need to think hard about, well, what type of immune cell are we going to use? Vast majority of immunotherapies that use cells at the moment use the commonest type of T cell in the body. T cells are a type of cell that usually fights viruses, and they're called alpha beta T cells. And the difficulty with that is that if you take alpha beta T cells from one person and give them to another person, you get something called graft versus host disease, where the donor's cells attack the recipient and they cause a lot of inflammation. Now, that's a problem because it means that if you're making an immunotherapy from that type of immune cell, you have to use the patient's own cells. And that's really expensive and takes about 3 weeks. So, a patient's disease might get worse during that 3-week period. There are other types of immune cells, such as gamma delta T cells and NK cells, that don't cause that graft versus host problem, which means that you can make them beforehand from cells provided by a healthy donor who may not be in any way related to the patient receiving the immunotherapy, which means that you could potentially make them, freeze the cells down, and then deliver them as soon as you wanted to when the patient actually needed them. So we use gamma delta T cells, and this is one of the reasons why, because they potentially allow you to do that. And they have already been safely given from unrelated donors to patients without causing big toxicities. So that's, I think, that has really proven the point clinically there. So, what did we aim to do? And this is, harking back to the work that was funded very kindly by the Little Princess Trust through the CCLG. But I'm also going to bring in some of our wider work, and we wanted to look at, firstly, if we're going to get the cells from the blood to the tumor, how can we keep them healthy along the journey? How can we make them get to a bone tumor and remain fit enough when they're in the bone tumor to deliver that kill? And if they're going to get there, what is it that osteosarcoma produces that we might be able to use as that homing signal? And how can those natural homing properties be enhanced through the engineering that we want to deliver? So it's really, to use the analogy that I've given, it's the steering and engine parts that we were really focusing on, though I'll talk a little bit more about the cargo part at the end. So a little bit more about gamma delta T cells, because they are a pretty unusual type of cell, and they're relatively sparse. It's a small subset of immune cells. Everyone has them, but they comprise only about 1 in 50 of your sort of circulating T cells, so you don't have very many. They are really, really ancient from an evolutionary perspective, and to the extent that some really very primitive organisms have gamma delta T cells, and they're much more ancient than the more advanced, kind of adaptive alpha beta T cells. As I said, they can be safely given from one person to another, so there is the potential for using them as an off-the-shelf therapy. Importantly, for one of the ways that we are using them, they're really, really good at killing target cells, so that could be cancer cells, that have been coated in antibody. So antibodies are another part of the immune system that usually stick to foreign or harmful things like bacteria and viruses or cancer cells. And gamma delta T cells can see when things have been flagged by an antibody sticking to them, and they're really good at killing those targets. And then the last bit, and this is important for this talk, because we're talking about bone cancer, that a particular type of drug called a bisphosphonate, of which zoledronic acid is a well-known example that a lot of patients with bony disease receive, incidentally sensitizes cells that see it to killing by gamma delta T cells. So you've got this opportunity to really enrich a sensitizing agent in the bone, which you wouldn't be able to necessarily do outside the bone, although at the very end, I will mention some ideas about how we could go beyond bone with this therapy. So first bit about the steering. How do T cells get to where they want to go? So T cells have these receptors called chemokine receptors, and it's a little bit like a nose, and they sniff out where the chemokine, which you could imagine as being a little bit like a smell, is coming from. And then they move towards the higher concentration of this chemokine. So as you can see, the chemokine receptors are on the front, and you get this kind of leading edge that forms. And, cancer cells, we know, produce chemokines, and the chemokines may come from the cancer cells themselves, or they may come from the immune cells that are sitting in the cancer. But this is a phenomenon which is fairly well recognized, and one that I thought we could harness within the context of osteosarcoma. So, okay, does that make sense? So we first looked at what osteosarcoma produced that we might be able to use to guide these gamma delta T cells. And this data was actually data that was generated in, I believe, in a Chinese lab, and it was already published, and we just reanalyzed it. But the point here is that, so this graph, everything in the purple box is something that is a chemokine that is consistently produced more in the cancer than in an adjacent piece of normal bone. And this study used samples from many patients, and they did paired tumor versus normal bone and looked at the expression of all of these different genes, and we filtered out the chemokine. So we knew that everything that was in this box was kind of where we wanted to be looking if we were going to look for what we could sniff out and what we could educate our T cells to be able to host. So we had a look at that, and this is work that's been done by Alba Southern, who is a PhD student funded by the Little Princess Trust on this grant. And we looked at a number of these different chemokines, and we looked at a number of different types of osteosarcoma. So these two are cell lines that you can purchase. These three are patient-derived osteosarcoma lines that we obtained through a collaboration with Katia Scott-Landy at the Rizzoli Institute in Bologna. And it was already known that if you make osteosarcoma a bit more inflamed, you get more chemokine, which is helpful because we want to do immunotherapy. We want it to be inflamed. So we provided an inflammatory cocktail and added either none, 10 nanograms or 20 nanograms, and looked at the amount of chemokine that is being produced. And as you can see, in general, the more inflammation you get, the more chemokine you get. Now, that's all well and good, but how can we make that useful? So the thing that we need to be able to do is see where the immune cells are going. And this is one of the ways of doing it. So what you do, you have this little slide here, and this is it's a kind of about 10 centimeters long by a few centimeters wide. And in it, you have these little chambers. And what you put a high concentration of your chemokine in this box and a none in this box, and then you put your cells in the middle, and then you watch them with a microscope to see which direction they move in. So as you can see here, if you have no stimulus, they just bop up. Each line is the cell, and this is an individual cell that's been followed with sort of serial photographs. And as you can see, they just kind of bop about, and they don't do very much. But if you provide them with a stimulus gradient, they all move towards the thing that is pulling them. And you can do this in a reproducible way, and you can graph it out with a measure called the forward motility index. So basically, how much directional movement are the cells doing? So we thought, okay, so we know what we want to move them towards. We know that we can measure it. So how are we going to do that? So we evaluated what chemokine receptors gamma delta T cells have normally, and we evaluated what movement they have normally, and then we gave them extra ones. So we engineered them to have more chemokine receptors, and we picked the receptors that correspond with some of the chemokines that we know osteosarcoma produce. The hypothesis was, if you put that on, they will move towards the cancer better. And they do. So using as an example, this receptor, which is called CXCR6, if you have supernatant, so that's if supernatant is the bit in when you're growing the cancer cells, it's the liquid that sits above the cancer cells. So it's what all of the stuff that the cancer cells make is pumped out into. So if you take some of that and you use that as your stimulus, as you can see here, the cells expressing this extra receptor do move substantially more towards the stimulus than cells that do not express. So these are unengineered, and these ones are just expressing a cytokine. No, I'll come on to that in a second. So I think we have demonstrated that it is possible to enhance the movement of the gamma deltas towards this osteosarcoma supernatant. Now, that's all well and good looking at it in the context of these little slides and watching individual cells, but tumors form lumps, and it's much nicer sometimes to see it in a larger context. So for that, we make these three-dimensional models of bone tumors. These are a few photos of different versions of that with the kind of explanation underneath. So in the black and white, this is the tumor, and this is a disc of tumor cells embedded in some collagen. So it's in a kind of stiff jelly. And these bits that look a little bit like solar flares, this is the cancer invading out of the original mass into the surrounding area. Here, what we've done is we've used green gamma delta T cells, and you can see, unfortunately, the collagen, it sort of fluoresces a little bit green anyway, so this ends up looking quite bright. But all of these gamma delta T cells here are ones that are approaching the tumor boundary and moving towards the tumor. And then what you can also do by using red tumor is look at the spaces. So once the gamma deltas have gone on and killed bits of the cancer, they literally carve holes in the cancer mass, and you can actually quantify the size of these holes in order to work out how much of that tumor in the 3D model has been killed. And this is just some examples of what that looks like. So the invasion of the tumor into the surrounding, which is normally a kind of relentless process, if you add our engineered cells that home towards the tumor, as you can see here, they start to invade, you add the cells, and that invasion stops, and you can control that. Now, what's really important, though, is if you add those engineered cells with the sensitizing agent, so this is with the zoledronic acid that I mentioned at the start, you can actually start to make that tumor shrink and make the invasion regress. And this is really key, because when we're doing immunotherapy, it isn't just about a one-shot system. It's about combining everything in the correct way. And this is something that people who've been designing chemotherapy protocols have been doing for years and years and years. But in immunotherapy, that has lagged a little bit behind, and people have, there's a lot of work has gone into thinking that delivering just the one immunotherapy is going to solve all the problems. But actually, the immune system is more complicated than that, and we may need to have a multimodal approach. And this is an example of how that could be harnessed. Again, as you see here, in terms of tumor destruction, you can see that the fully engineered system with the zoledronic acid gives the best kill compared to any of the others. So it's really kind of leveraging the multimodal approach that we're using. Okay, so we know that we can get the cells towards the tumor, but you have to keep them healthy on their journey. And this is particularly important when we're using gamma delta T cells. So other types of T cells are quite good at being fairly self-sustaining, but they cause graft versus host disease and so on. So it would not necessarily be what we wanted to do. Gamma delta T cells are less good at being self-sustaining, and they get essentially addicted to the stimulus that you use to grow them up in the lab before you would give them to a patient. So this is what happens. So IL-2 is one of the stimuli that we use. And as you can see, you expand them in the lab. And if you deprive them of IL-2 after, in this case, day 7, they just die. Whereas if you keep on giving them IL-2, they continue to expand. Now, this is a real problem because, patients don't come filled up with IL-2 in order to be able to keep your given cells nice and happy and alive. So if you're going to give these cells to a patient and they don't have all of this IL-2 going around, well, then the therapy is not going to work because the cells just won't persist. So we had to find a workaround for this. So what we did was we chose a different stimulus called IL-15, and we made the gamma delta T cells produce their own IL-15. So they're essentially producing their own food. Now, this is something that means that you don't need to continue giving them IL-2. So it's the same experiment as I showed before, but as you can see here, the cells, even after the IL-2 drop, the cells that were engineered, so either this one called IL-15 or this one called STIL-15, I'll come on to the difference between those in a second, they continue to proliferate. And we've also shown that they will persist in mice, because we do a lot of this modeling in animals to start off with, and they have a much better cancer-killing profile than the unengineered cells. Now, as I said, we do two different forms here. So the IL-15 form is just the normal form of IL-15. And when we engineer the cells to produce that, they produce a little bit. It's consistent, but there's really not very much. So what we did was we got IL-15 and we stuck an extra bit on it. And it's actually a little bit that comes from the receptor that detects IL-15. And so we called it STIL-15. And as you can see, you get a vastly superior quantity of the detected IL-15. And it is also, other people have done this, used a similar approach. It is also more potent as a stimulator of that receptor. So I think in general, it's certainly much more helpful. And you see here that not only helps the gamma delta T cells stay alive, it also means you can recruit other cells in the immune system. So rather than relying entirely on the cells that you give to the patient, you can also have a benefit to other bystander immune cells that can also be activated, that can also help attack the cancer. So it's about recruiting all of the help that you can get from within the immune system rather than relying entirely on the engineering that you're delivering. Again, that's a differentiation from conventional CAR T approaches. So this is what we've got. We've got these unmodified cells, unmodified, they don't like it, they die in the absence of support, they don't activate bystanders, and they kind of fall apart. IL-15 armored cells, they stay happy, they don't produce a huge amount of IL-15. The NK cells aren't so activated because it's in not quite low quantities. But with the STIL-15 version, there's lots of it about. It's very potent. You activate the gamma deltas, you activate the unmodified gamma deltas that are the bystanders, they stay happy, and you also activate the NK cells, really maximizing the number of, essentially the number of soldiers you can throw against the tumor from within the immune system. Now, that's all well and good, but and this is the kind of the last section, you really need to be able to deliver that kill signal. So this is coming on to the cargo part at the end. And I've already discussed what antibodies do. They stick to targets and mark them for immune killing. They're an incredibly important part of vaccine responses, incredibly important part of the immune system. And, they've been shown to be active against cancer. So patients with neuroblastoma, for instance, get treated with anti-tumor antibodies, and that has been shown to be quite effective. So this is something that we wanted to leverage. Now, I've already said that gamma delta T cells are good at killing antibody-coated targets, so we made them make their own antibody. So here, this is the approach, and this is something that the paper on this came out last Wednesday. We take a healthy subset of immune cells, these are the gamma deltas from a healthy donor. We genetically engineer them to produce two things. They produce this antibody, and they also produce this STIL-15 cytokine. And that means that not only do they have extra anti-cancer power because of the antibody, they also stay alive to keep fighting because of the STIL-15 and recruit other cells that can use those antibodies to also attack the tumor. Now, the question is, does this work? Yes, it does. Certainly it does in animal models. So we wanted to test this against osteosarcoma. So what we did is we got some patient-derived osteosarcoma samples, the same ones that I showed you before that we got from our collaborator in Italy. We put them in the tibia, so it's a leg bone in mice, and you can detect them using a special camera. This is it glows when you detect them with this camera, and then we tracked the growth of those tumors after treating the mice with various different versions of the therapy, and this is what we saw. So as you can see, so the gray line are mice that received unmodified gamma delta T cells. And as you can see, they don't do very much. This looks essentially exactly the same as completely untreated animals. And we wouldn't expect unmodified gamma delta T cells to work because they don't persist in the animal. If you engineer them to persist, they maybe do something, but it's really not very much because they aren't given that extra anti-cancer power. But really importantly, CAR T cells, which have been lauded in the context of hematology, also did basically nothing in this animal model. We tested them beforehand, before we put them in animals. They were powerful CAR T cells. We knew that they worked. We knew that they killed osteosarcoma, but you put them in the animals and they don't work. If you take these armored gamma deltas, so the ones that produce the STIL-15, and you add some zoledronic acid, so then you sensitize the tumor to killing, suddenly you get this enhanced survival. so significantly enhanced survival. This is comparing the green line with the blue line, and it looks like we're starting to make a bit of a difference. But really, the killer is that if you add in the antibody as well as the cytokine and the zoledronic acid, you can really get to up here. And, all of these mice were doing pretty well. And if we actually look here, so each line here is the growth of a particular tumor. So it's an individual tumor in a mouse. So unmodified, cytokine only, cytokine plus zoledronic acid, CAR T. But really, it's this. So, there's only one mouse whose tumor really escaped at all, and the rest of them are pretty much lower signal at the end than they were at the beginning. And this was out to 90 days. We terminated the experiment at 90 days because we'd really reached the point where we felt that we were happy that we'd seen the result. So that's what we've been doing. And so we've been guiding the cells to the cancer, keeping them healthy on the way, sensitizing the cancer to the killing, and then delivering that payload. Now, that's all well and good because in the context of tumors that are in the bones, because the zoledronic acid binds to bone. Now what we want to do is look at tumors that have escaped the bones. We know that sarcomas metastasize to the lungs, for instance, and I've now in the process of submitting some applications to look at other sensitizing agents that we can use to hit disease that has gone outside the bone. And I'm quite pleased that Will from the Bone Cancer Research Trust is here, because at some point you might see that crossing your desk. I think the deadline is in about 2 weeks. So we'll see how that goes. So yeah, that's what we do. I'm happy to take questions, and thanks very much for putting up with me talking for the past half hour.
Well, thank you very much for your presentation. It's really interesting, and I really like the Lego. I like anything that includes Lego, so I think it's a great analogy. We have already had 3 questions. So
okay. How do I see them? Do I just click on the Q&A tab?
Oh, I normally read them out.
Oh, right, okay, cool. That's fine.
Do you want to stop sharing your screen if that's okay, and then everyone can see our faces? Okay, so the first question is, why does this approach work in solid tumours where previous attempts at using T cells have failed?
So I think it's because we're not just doing one thing. So previous attempts using T cells have typically relied on a single piece of machinery. So for instance, a CAR, so in CAR T cells, if this CAR T is a single switch, it only really benefits the cell that has been engineered. So the other engineered cells in the bag don't really get a huge amount of benefit. What we're doing is it's a multimodal platform for delivering immunotherapy. So the gamma delta T cells, all gamma delta T cells have anti-tumor activity naturally. We're enhancing that further by sticking the antibody on the tumor, enhancing it even further by providing the cytokine, and then enhancing it even further by sensitizing the tumor cells in the bone using the zoledronic acid. So it's about embracing the fact that the immune system is very complicated and it is naturally multimodal. So therefore our approach should be multimodal if we want to manipulate it. And that's why I think it's worked so well.
Okay, so second question from the same person is, if the chemokines are also expressed by the non-cancerous cells, is there associated side effects from these?
So we haven't evaluated the chemokine receptor engineered gamma deltas in a mouse model yet. So that ultimately we don't know. What I would say is that gamma delta T cells typically do not target healthy cells because they have a sort of innate cancer detection mechanism. So whilst they may go to an area where there were healthy cells, it wouldn't mean that they would then attack and kill the healthy cell. The cancer specificity and the attack and kill bit is on other modules that we put in to ensure that the therapy can be given in a safe way. So, for example, the things that you stick the antibody on you make sure that that is something which is highly expressed in the cancer and ideally not highly expressed in the healthy tissue.
Okay, so next question is, what is the cost of this immunotherapy? They did add another question about the lasting side effects, but as you said, you've not quite assessed that just yet. Things take a while, don't they?
No, immunotherapy, so cellular immunotherapy in general. So to date, immunotherapy that has been made from the patient's own cells typically costs the NHS around £250,000 per patient. So it's very expensive. The advantage of being able to make immunotherapy from healthy donor cells is that you can make a lot more and then use one donation to treat multiple patients. So the cost, it would be substantially lower than that, simply because you can manufacture it at scale and you can manufacture it from someone who has a really healthy immune system rather than having to use cells from a patient who may have had chemotherapy beforehand and so on. So their immune system and their bone marrow would have been hit by whatever they'd been treated with before. So the anticipation is that this is really going to be a much more cost-effective way of delivering immunotherapy. Now, it would be impossible to put a figure on that at this point because we haven't done it yet. You know, we're hoping to do that, and we're actually working out the at-scale manufacturing processes for this at the moment. So watch this space.
So would you expect that the gamma delta T cells could come from like a normal stem cell donation type thing?
Yeah, so the you can get them from a standard kind of leukapheresis or apheresis, and it's something that healthy people can have. And you know, actually, there are companies that will pay people to donate cells for use in research or in clinical trials, and it's something that's actually a service that we use in order to get the cells that we use in our experiments.
Yeah, that there's already
there's already a pipeline to get it. Yeah.
Yeah. Okay, so we do have more questions. So somebody would like to know, what does the antibody target? Is this co-delivered in the same vector as the STIL-15?
So yes, so it's the antibody that I showed here. It's an anti-GD2 antibody. So it's the same. It's essentially the same antibody. It's not quite the same, but it's the same antigen recognition part as is on dinutuximab, which is the antibody that is used for neuroblastoma. It is in the same construct. It's in the same plasmid construct as the STIL-15. If you want to see the specific molecular bits about it, the paper's in Science Translational Medicine from last Wednesday, and all the details are in there. I can go into the technical details now if you want, but I suspect you might not want me to.
I can put a link to the paper in the email that asks for feedback.
Cool. Okay.
Okay, so next question, which is slightly technical. Well, they're all slightly technical. In the in vivo study where you compared the results with the CAR-AB T cell, was this done with the same donor as the gamma delta T cells?
Yeah, so same donor and also same targeting moiety. So it's exactly the same antigen binding motif. It's just on a second generation CAR format rather than on a as secreted as a synthetic antibody.
Okay, so next question is, can you tell us more about the potential for combining the T cell approach with other agents like chemotherapy?
So that's been tried in and is something that I think is potentially quite helpful. But I think that the thing that we need to make sure is that you don't end up shooting yourself in the foot. So chemotherapy kills cells that are rapidly dividing, and it kills immune cells. So you wouldn't probably want to give a load of immune cells that you've however expensively engineered to your patient and then give them a load of chemotherapy that was going to wipe out those immune cells. Having said that, you have to use some chemotherapy to prepare the patient to receive the immune cells. So there is a kind of combinatorial effect. What I envisage immunotherapy helping for is really for mopping up bits of disease that chemotherapy has not been able to touch. So one of the things that cancer does is it divides rapidly most of the time. Those cells grow rapidly most of the time, but sometimes they go quiet. And when they're quiet, it's very hard for chemotherapy to hit them. Now, if we're going to talk specifically about sarcomas, we use surgery to remove the big lumps. And then, there's potentially little bits that you can't see on a scan, that you can't see kicking about in the body with other imaging, that may then lead to the cancer coming back in the future. And that, I think, if we were going to have to be in the ideal position to give this to patients, that is where I would put it. Difficulty, of course, is that you have to test it in other settings first before you can get to the ideal clinical setting that you want to test it in, simply for safety reasons, because obviously we've never given this to patients. And we need to make sure that it's safe first before I go and start recommending giving it to people. So
makes sense.
Yeah.
Okay, so a very relevant question, actually. That was asked quite a few minutes ago, but it's very relevant to what you said. What challenges do you envision with bringing a complex therapy like this to the clinic, particularly for pediatric patients? Then they added encompassing the cell therapy plus cytokine plus antibody.
So I think there are two challenges. Well, there are lots of challenges, but they come into two categories. There'll be the logistical challenges, as in how are you going to be able to make it and have the clinical pathways and pipelines in place to be able to deliver this type of therapy. And that is very well established in hematology, because leukemia patients have been receiving modified cell therapies for a number of years now. So a lot of the expertise for how to make the cells, how to get them to the patient, how to thaw them out, how to deliver them, how to provide the supportive care to the patients who are receiving them, that actually already exists. And there are trials opening, at the moment where we'll be giving modified immune cells to patients with solid tumors. So, that is something that is already happening. Then there's the other side, is like, well, how are you going to be able to justify giving this more complicated immunotherapy rather than a simple immunotherapy? Well, that really is to do with liaison and discussion with an organization called the MHRA. So that's the Medicines Health and Regulatory Agency. And they evaluate whether you can justify doing what you say you're going to do. And, we have already met with the MHRA once, and they didn't think that we were completely crazy. So that was quite helpful. And, I think it is about building up a body of evidence to justify what it is that we're doing. There will obviously need to be a very cautious introduction of anything like this to make sure that it doesn't cause unexpected side effects. But there are protocols and procedures in place for doing that. And I'm quite confident that the infrastructure for delivering this type of therapy will evolve. Excuse me. Certainly at UCLH, we are very much growing our infrastructure to deliver this. And I think it's something that will be deliverable in other centers as well. I mean, certainly in Great Ormond Street do it frequently and have been doing it for years. So, it isn't in the realms of fantasy.
It sounds really promising. So I'd say I have 4 more questions, but it's actually 3, because one of them is to say congratulations on publishing your paper.
Thank you.
I've got one saying, do you have any plans to trial this with other types of bone cancer?
Yes. So one of the things that we're really interested in doing, there's a number of different things. So firstly, this doesn't necessarily only apply to cancer in children and young people. So if you think about, cancer that starts in the bones is kind of what we're talking about here, but there's also cancer that has spread to the bones. So that could be breast cancer, prostate cancer, kidney cancer, lung cancer. All of those things are potentially targetable with this type of therapy. Might not be the same antibody, but if you use a different antibody, you may be able to hit a number of different cancer types. So we definitely have plans to look outside the pediatric space and outside the TYA space, and also to look at adult cancers. And then secondly, other types of bone cancers. So yes, we know that gamma deltas can target Ewing's sarcoma. We know that the Ewing sarcoma expresses GD2. So that's one that we're going after. Will from BCRT, please take note. And we know that you can hit the tumor in the bones. We think that by changing the sensitizing agent and using something else that doesn't bind to bone quite so strongly, we can hit tumors that are not in the bones, and that's something that we want to test. So yes, we are looking at a number of other settings.
Okay, so I wonder, I've had another question. Are you okay with 3 more questions?
Yeah, I'm good. I'm good.
Okay, so do the T cells live after they kill the cancer cells, and how do they protect from the cancer reappearing?
So they do live after they've killed the cancer cells. We don't know how long they would hang around in the patient because we haven't tried it. I suspect that unlike immune cells that are made from the patient's own cells, they will probably not hang around that long because eventually they'll get rejected by the patient's own immune system. But I think the important thing here is to not fall into the trap of assuming that solid cancer is the same as leukemia. In leukemia, there's a lot of data that says that if you need you need to have that long-term protection and that long-term persistence of your immunotherapy to prevent the leukemia coming back. But you can't treat leukemia surgically. So, in sarcoma or in solid tumors, we have this option of taking out the bone cancer, and you take out the lump. It's the standard of care is surgery. So you've got other weapons to be able to use in the management of that cancer. So the long-term persistence might not be so much of an issue. But we have to wait and see. Ultimately, we need to see if that works or not. I saw a comment in the chat which may have related to that. Yes. I'm not sure what is that IL-2. It just says IL was used in the Euromos trial, but I wasn't. I don't know which IL you're referring to.
If you want to drop a bit more info.
Oh, interleukin. But is that so? I mean, I think if that was interleukin 2, then yes, interleukin 2 does give you quite nasty side effects, and it is something that is a real issue when you administer it in high doses to patients. We wouldn't advocate doing that. That's why our cells produce their own cytokine, and precisely it's not IL-2, it's IL-15. And it's in much lower amounts than you would get from the sort of dose that you would give to a patient. Furthermore, as I said before, when I was talking about giving immunotherapy alongside chemotherapy, if you stimulate the immune system a lot and then you fill a patient with chemotherapy, you make all the immune cells expand, get really activated, and then you kill them with your chemotherapy, and then you do it again, and then you do it again, and understandably that doesn't work. So you need to either use the immune system or use chemotherapy and work out the timing between the two. And we know a lot more about how you can manipulate the immune system now than we did when Euromos was originally run. And similarly, zoledronic acid, which I talked about, was used in combination with chemotherapy in the OS 2006 trial in France. and they showed that it didn't really provide any benefit. But then they weren't using it as an immunomodulator. They were using it as a bone strengthening agent, and actually it wasn't as good. But that wasn't because of its immune effects. That was most likely because of other effects, because they were giving it alongside chemotherapy.
Okay, so last two questions. What was the reason for using a secretory antibody versus a chimeric antigen receptor in the gamma delta T cells?
So we can do chimeric antigen receptors in gamma delta T cells. That's no problem, and I've done that before. The problem is that a CAR is stuck in the cell membrane, so only the cell that has been engineered receives the benefit. What we wanted to do was acknowledge the fact that there are lots of other cells that may well be present in the vicinity of the tumor that can also engage antibody-coated targets and can also attack the cancer. If you give them an antibody that is secreted into the vicinity of the tumor, you empower them to do that. Whereas if you just go in with a CAR, they just sit there. They don't get any benefit. So you're not using the resources that the immune system is providing for you.
Okay, so last question is from a parent whose child is currently having chemotherapy. So they said it was very informative and interesting. They've asked if there are any papers you'd recommend to a layperson to learn more about this subject.
I'm just trying to think. Depends how much extra kind of immunology reading you want to do, because a lot of the papers on this are written very much with a scientific audience in mind. There are some more accessible reviews, and I'd point you to a journal called Frontiers in Immunology, which tends to do quite accessible review articles for people who are not so specialist, and that would certainly be somewhere that you could find out more about particular parts of immunology. I think the important thing to say here is that what I've talked about here is that this has not yet gone into patients, and even with the best funding in the world, this would probably not be in patients before, I would say, early start of 2026, because you've got to go through all of the processes to make sure that you can do it right. And I think we want to get this right if we're going to do it. So I wouldn't worry too much about learning about this specifically, because it will probably have moved on a little bit by the time it actually gets to when it can be clinically used.
I have to say, I didn't expect that it'd be quite as soon as 2026. I was thinking
what we're hoping for.
Drug development of like 10 plus years. So I mean, it sounds fantastic, really, especially if it's going to be helping people soon-ish.
Hopefully. I hope 2026 wasn't over-promising, but that's what we're aiming for.
I'm sure everyone understands that these things are complicated. So that is officially all of the questions. So I've got a little bit to read, but I don't know if there's anything you'd like to add beforehand.
No, just thank you. Thanks for the opportunity to present our work. Big thank you to you guys for helping fund it, and to Albert and to Dan, who you kindly paid the salary for actually doing the work. So they've done an amazing job. And yeah, I look forward to hopefully updating you with more findings.
I look forward to hearing them. So thank you very much for joining us tonight, everyone, and thank you very much to Jonathan for your insightful talk. So it has been recorded and we will be sending it out in the next few days if you'd like to share it or watch again. You will also be receiving an email shortly asking for your feedback on today's talk. This is so we can make sure our talks are the very best they can be. So please do take a couple of minutes to let us know what you thought. And have a lovely evening, everyone. Thank you.